That means we start to sing. First of all, I want to commend all of you here who went right through the ick factor about the fact that this is a talk about worms. I'm Dr. Jon LaPook. I'm the chief medical correspondent for CBS News, and I'm a professor of medicine at NYU Langone Health. I do internal medicine and gastroenterology. Talk about your coincidences. So the ick factor means nothing to me. This is Dr. Andrea Choe, and she was going to introduce herself.
Well, you just said it. My name is Andrea Choe, and I'm the CEO and co-founder of Halflife.
So I'm so excited about this because I think there's philosophy here and then there's science here. And the philosophy is sort of, you know, we've been around for millions of years, and we think about, you know, all the advances in science, and when we get infected, we just wipe out those infections, you know. It's like, and it's worked out for us a lot in the past. Childhood diseases and immunizations. If you get pneumonia, if it's bacterial pneumonia, you treat it with antibiotics. And if you get meningococcal meningitis and it's bacterial, you give antibiotics. So there's a lot of good things to say. But Andrea, who started off, her story is so fascinating because she started off doing research. I think in the made, if everything goes right, in the made-for-TV movie about this, all of you are right here, kind of towards the beginning of it. There's going to be a long journey ahead, but it's going to be very cool for all of us to follow this story because she found something. I'm going to set it up for you to sort of give the background. She found something in sort of she's an MD PhD. She'll tell you about her training, but she found something. Never go away on vacation. Her advisor went away on vacation. And while he was away, she turned her research from this to that, and she found some fabulous stuff that led her on this amazing and really interesting road. So with that as a setup, and then we're going to go back and forth, and we'll talk about, we'll try to stay away from med speak, but I think it's a fascinating story.
Oh, thanks. Now I can't stop thinking about my made-for-TV movie. I just want, what love is Aquafina.
Okay.
You know?
Aqua.
Okay.
Aqua.
Right.
Put it in the calendar.
Okay. Well, you know, just like you said, when I started my PhD at Caltech, so when you go into a program like the MD PhD program in the U.S., you do your first 2 years of med school. So you've learned, you know, done all of your book studying on what diseases are and how they look. And then you choose where you're going to do your PhD. And so I went around Caltech. They have amazing labs there. There's one worm lab there. And in this worm lab, with a PI being Paul Sternberg, I went into that lab and I just was in love. Because first of all, worms, I know that you think about earthworms as the ones that come out the sidewalk after it rains. Those are the segmented worms. I'm thinking of the I'm thinking of the roundworms. I'm thinking about actually have one body container. So like if you poked it, it would pop, whereas a segmented worm, it would not. That's just one, I guess, icky way of thinking about it. But the roundworms. So if you go into a roundworm lab, the first thing you'll notice is that people are sitting on their benches with microscopes, and there's a little agar plate, and they're picking worms with this piece of platinum, really expensive, by the way. And then we would get the platinum and pick up a worm and move it, and I would see what people would do, because with roundworms, you know one cell from when it's an egg to the 1,000 cell of a whole adult body. Every single neuron has been mapped. Like someone a couple of decades ago did cryo-EM and stitched together every single synaptic connection in a worm. So you can study anything, any gene. You could thaw any mutant you want from a freezer of worm mutants. And
How many genes do they have?
They have, that's a good question. I don't
I'm
remember
sorry.
how many.
That wasn't a gotcha question, no, but it was like, because we have like 30,000, anybody out there? We have like 30,000 genes or something, I wonder how many they have that. But the point is that they're much simpler.
I can't remember exactly how many genes they have, but you know what I do remember is that a lot of the genes are actually similar to the genes that we have. So it's really interesting because they are a model organism to study life in so many ways. And so the time that I went into Caltech was also 5-6 years after Human Genome Project. And so now we had all these genes we were interested in. It's like, how do we study it? Oh, well, we can use worms to study that. We can knock out the gene and see what happens. Or we can let it live and then feed it a bacteria that'll knock out a gene when it turns like 3/4 through its like life stage, and then figure out what happens. You could study anything. So anyways, that was why I went into worms. That's a question that people always ask me, like, how did you get into worms? So then me and my PI came up with this great genetic screen project, and then he went off on his sabbatical, and then I just fell in love with something else about the worms. I was like, wait a minute, we know so much about what happens within a worm. What about the stuff that's happening between the worm? And also, this is a time where Caltech had brought in a lot of speakers about quorum sensing and swarm behavior.
What is quorum sensing?
It's how animals communicate. They can take a vote, like bacteria. Okay, Pseudomonas aeruginosa, right? You know that this is one. It's a bacteria that there's a lot of nosocomial infection.
Right. Nosocomial meaning it happens in hospitals by our doing.
Yes, so people can get infected with Pseudomonas and in their lung, and they can wait and stay quiet until they take a vote and they say, are there enough of us here to release our toxin at once? And that's what they do to create damage. So people are trying to interrupt that sort of cycle by interrupting the way that they communicate, worm sensing. But also birds in the sky, how do they swarm? The fish in the sea, how do they swarm? I can't, if someone tried to get me to be that coordinated in a dance troupe, like, forget it. So it was just so interesting.
You haven't done synchronized swimming, obviously.
No, I am so impressed with synchronized swimmers, so impressed. But so that's what I was really interested in, is how the worms were communicating with each other. They don't have eyes, they don't talk, right? So there must be some pheromonal molecular language, and it's controlling all kinds of interesting things. How it's going to sleep, how it's even longevity. You know, how we know that we're studying worms to understand aging, and how they're mate finding, how they're moving towards something, away from something, knowing where there's predator prey. So anyways, that's what I was interested in. Now, where my project turns is that I discovered this pheromonal molecular language between different worm species that we studied.
And then my job, should I decide to accept it, pheromonal. Just for people who it's kind of a word we hear, but what do you mean by a pheromone?
It's like a chemical signal that's produced by a cell that communicates with another cell to do something. You have pheromones that are secreted by your body that's also controlling like hormone regulation and whatnot. You might be attracted to your partner and you don't know why. Maybe the way they smell, right? So anyways, I think that there's a lot that, you know, we were studying with this molecular language. And what we found was that the language that we discovered around one species of worms was actually the same one that was being used by a species of worms that had diverged hundreds of millions of years from it. So think about that, like compare that a worm that was recently evolved to one that came on the earth around the time of T-Rex, right? That's a long time apart, but they were using the same language. So that struck me. That was the first thing. Which is, how is it that they're using the same language? The whole point is you kind of want to use different languages so you don't get confused.
And when we were talking beforehand in a Zoom call, it's amazing. I mean, across the world, there's all these different languages, right? Peter Lee is here. How many different computer languages are there, from assembler, Pascal, on and on, thousands and thousands? What was it about this that made it preserved so that it's the same across, you know, all of these different worms? So that's amazing.
I don't know. Right? I still do not know. That's something that, you know, even if we had a time machine and went back to that time, we still would not know. But, you know, we found the same. For example, there's I think we got an apple that fell from a tree somewhere in Paris. So romantic right now. We found worms from that, and also one from like an intestinal parasite, and they were the same language. They would never see each other in modern day.
Right.
So, you know, that was just really interesting.
And then to give people an example, I'm so sorry to interrupt, but if you think of a tree with different branches going out, and you could change things along the branch, and you have a lot of diversity. But there's something about this, I mean, it's a really important point for your research, and I want to make sure everybody gets this, is that this was at the base of the tree. So this was so basic that if you changed it, the tree dies, I guess, or something very important changes. So that obviously was inspirational for you in the story you're about to tell.
Yeah. Well, we don't want anything dying here today.
No,
So there's that.
but certainly no trees.
Yes. And I think that the thing that where the project turned from taking what you were saying in that discovery and that core preserved language is that I heard this Radiolab podcast where I heard about this guy that had intractable asthma and nothing would help him. And he heard about how for some reason if you have gut roundworms, you are protected from these diseases. And he went to Africa and walked barefoot to get hookworms to then inhabit his body, and it worked, and he came back and he tried selling it in the US, and of course people shut him down, but he's still selling it out of Mexico. And so I was like, wait, what is going on there? So then after that Radiolab podcast, I started reading more about just overall the phenomenon of where did that philosophy come from? And so, like I shared yesterday during my big idea, if you look at the world and you ask what countries have the highest incidence of allergic and autoimmune disease, US is number 1 and number 2. And sub-Saharan Africa has actually got the lowest incidence of those diseases. And it's kind of remarkable because why? Was it lifestyle, genetics, diet? And what we know is that they still have gut-dwelling roundworms. So that's epidemiological.
And the question, as we talked before, was, is it true, unrelated, or is it true, related? And that's this part of the story that she's going to be telling.
Yeah, so I'd say there's decades of work already being done by amazing worm scientists all over the world, where they took worms and they put them in animal models of diseases, and it helps to block disease. And so I kept reading and reading and reading. It was like, wait, it's blocking asthma, MS, diabetes, multiple sclerosis. It was the span of all these allergic and autoimmune diseases. And then people would take secretions, and then, that would work too. And then I would say, okay, then what? And that's where they would stop. And I was like, why? And then I would go to the other paper, and I'm like, okay, what is this author now doing? Okay, they're in another lab, and they're studying something else. I'm like, why, right? So I think that's where I thought, wait a minute, let's study, first of all, let's go back to people are looking for what could it be that there's some common molecular chemical that's being secreted by worms, but more than one worm is working. Does anyone know what that common molecular molecule could be? Common molecular signal could be. And I thought, wait, I have it, right? Because I had just studied common molecular signals. So we took that, we tested it, and it worked. It was blocking multiple diseases. So that's
the moment in the made-for-TV movie where she's driving in the car. And then you're afraid because it's a close-up, don't get into an accident, and then it comes back and you have
driving in the car looking at data?
No, you're listening to the Radiolab.
Oh, right.
Listening to the Radiolab, and it's this moment where she goes, wait a second, I wonder how that works. I've been doing, accidentally or coincidentally, I've been doing research on that very thing. So that's the moment where chocolate met peanut butter and collided, and fabulousness.
Not even peanut butter and jelly, but chocolate and peanut butter. I'm going to have to try that one. I was thinking of a sandwich, like you would.
Reese's
thinking of your hands. I heard like 3 people whisper, like, what is wrong with her?
No.
Reese's.
The Reese's people out there, Reese's roundworms, think about it. Just consider it. Okay, sorry, go ahead.
So where were we? Now all I can think about is chocolate.
No, which is a good thing to talk about. No, you're talking about the fact that you're the person who actually, wait a second, I've already been doing that research. You isolated, you were looking at that pheromone, and so then go back now into, you've got this idea, you now know why it may be even more relevant than you ever thought. You were studying it because you said, I wonder how they talk to each other. And now it's like, wait a second, this could be a key to maybe doing something about autoimmune.
Exactly. So we took those molecules, we patented it, we tested it not just in animal models of disease, but also understanding if we were to ingest this, what would happen to it? Would it break apart? Would it last forever? We don't want to give anybody anything that's just going to stay in their body forever. How would this work as a drug? So then that's when we started the company. That's when I started Holoclara with my co-founders, one of them being Paul Sternberg, the head of the lab, but also an incredible metabolomics expert that was also involved in the project.
Metabolomics meaning?
The study of metabolites and all of the metabolites that are being secreted in this instance being worms. So, this was the 3 of us really started this company with an effort to not just take this molecule forward to help serve all of the millions of patients in need, but also figure out how can we actually mine more because no one else is. How can we go back and figure out how can we take all of this and create a worm atlas, and how can it serve many other things? So for example, one thing I didn't mention, if you look at the publications these days, you can see that there's worms and worm secretions that are also blocking obesity, right? And also they are able to promote wound healing. They're turning on intestinal stem cells. You know that in your intestines, if you took a picture of your intestines when you're younger versus when your intestines as you get older, the crypts of your intestines, the villi, they start to sparse out and they start to get shorter, and it gets harder to digest food, etc. And the intestinal stem cells live within the crypts between those villi. And worms are secreting things that are actually increasing the number of those stem cells. It's remarkable. And yet, what molecules are doing that? Can we identify them and take them across the clinical line? So that's what we're doing. That's an example of what we're working on.
And I want you to get into what those molecules are. Is it one big molecule? I know I read your paper, which is really deep in terms of the science of it, but I tried to understand what I could, and there are some specific molecules that you're studying. But I want to just take a step back just for a second and take a pause and say, why are the worms in there in the first place, right? I mean, they're in there, and we know what's in it for the worm, right? It's a nice, cozy place to live. There's food coming down. They don't have to go hunting for food. What is in it for the human host? So it's kind of like a symbiotic relationship. And one of the things that pops to mind, but I'd love to hear your take, is that for the worm, why is the immune system not just throwing it out? Why isn't our immune system just getting rid of it like it does with other things? Although we have a microbiome, right, which is trillions of bacteria and thousands of species that are in there, and it turns out they're doing a lot of things. But if it turns out that these worms are secreting something that sort of turns down the immune system, is that how they're not being expelled? Because that's how they're able to stay there? What's in it for us?
Yeah, so we'll just imagine, first of all, that you're a worm, okay? And
not a stretch.
Yeah, pick a century. Pick any century between now and whose host do you want to live in? Queen Victoria.
All right.
Okay, so imagine you're a worm living in a gut. Okay, and you have time, and you can secrete things and try them out, right? Trial and error. So what is it that you would want to secrete if you were a worm living in the gut of a host? One is the inflammatory response. You do not want your host's inflammatory response to be overinflamed, sending things all the time that's going to eradicate not just the worm, but also damage your host. So that's number one, is it can help to dampen that overreactive inflammatory state. That's helpful for the worm, it's helpful for the host. Number two is wound healing. Think about it, you want your home happy, not hemorrhaging. So if you see a wound somewhere, it's like you investing in spackle for your home, right? You want to do that. And also, who do you want to, sometimes when we get a wound, sometimes we send our own immune system to come fight it, and it gets worse. It's like sending the police force to come in and fix a hole in the wall, and then they start shooting it, and you're like, wait, no, you're making it worse, right?
So you need the immune system to do healing, but you don't want it too much, so you want to dial down to just the right amount.
Yes, and I think it's in the worm's benefit to find secrete molecules that can lure those reparative cells instead of the guns of blasting cells to come and repair that hole in the wall. And they've had eons of time to figure that out. And then on top of that, think about why is it affecting obesity? Why would it? Because it's caring about how its home is spending energy. It's like packing the fridge. You want to make sure you're home and you're packing food in your fridge and you're cleaning it out and continuing to do that. And then sometimes your immune cells that normally do that, they just get confused. They start packing the pantry over and over and over and over and over again, and you get obesity and you have inflammation.
Is it affecting absorption of nutrients in the small intestine?
Absolutely. So
the substances that you've been studying.
The substances that I've been studying? Does it affect the...
I mean, so we would think that the worms could do this, that, or the other thing, but what do we know?
Yes.
So
We
that's
know
a
that
good
worms-
amuse-bouche. I hope your bouches were amused by that. But it's important, I think, to get like the why of it. Like why? So now we sort of have an idea of like why there might be this relationship. But now bring us, because we have 15 minutes before Q&A, and I know there's a lot of that here. Tell me about your specific research, about what compound or compounds you've isolated, what you've done in animal models or whatever model, lab models, and then what you're doing going forward in terms of studying it in phase 1 and phase 2 and all that stuff.
Sure. So we're starting with the small molecules that we initially discovered. I'm a small molecule enthusiast. I will say proteins are great too, but proteins are things that are bigger, and small molecules are things that are smaller. When you think about a protein, it's usually larger than, they call it 3 kilodaltons as a molecular weight. And whenever it's that large, it's hard to administer to a patient because your stomach acid will break down the protein. Think about an egg. You expose it to heat, it goes from clear to white, and you cannot go backwards. It is now forever changed. So with proteins, we just want to be thoughtful. That's why people that take insulin, for example, have to get it injected, because if you try to take it orally, which is a protein, it would get denatured. And so I was always interested in small molecules.
And what category is it, and how big is it?
So we're dealing with small molecules for our first program. Is a small molecule.
How big are they, and is it I read your paper, but I want you to say like what it is.
It's a couple hundred molecular weight. It's kind of like sugar, glucose. It's kind of like that times maybe 1.5 to 2.
Mm-hmm.
So
what else does it have attached to it aside from the sugar base?
Yeah, so the molecular language is this unique sugar moiety that's not found by any other animal in the world. It's only made by worms and all kinds of worms. And then there's this carbon lipid tail where variations of it are like different letters of the alphabet. If it has a carboxylic acid versus an ester or a chain that goes this way, that's how worms are using to say this is this and this is that. So that's the initial family of molecules that we're working with.
Right.
And
there are different ones.
There are different ones. Right. And also they're orally available because they're not getting denatured by the stomach. Also, I think that once it goes in the gut, it's kind of intended, like how the worm would be secreting it on a daily basis. We're trying to reproduce as close to nature. Like, have you ever seen Jurassic Park?
Have I ever seen?
Do you remember Jeff Goldblum where he was just like, don't mess with nature?
Right, yeah.
Right?
Of course.
That's for our first program. That's really how I feel. I do not want to mess with things. I want to stick as close as possible to how the worm intended, how nature evolved it.
So
nature's already innovated.
So these substances are actually from the worm. You're getting them from the worm. It's not like you're getting them and then you're tweaking with
Originally, we don't have like worm vats or anything like that. We're growing a bunch of worms. Once we identify it from the worm, then we work with brilliant chemists. We're working with chemists at Caltech, Brian Stoltz, for example, and we make it from scratch.
Is it exactly what the worm made? Or
exactly.
So it's not, you're not adding a little this, a little that.
We have, but I think for our first program, we're not.
Gain of function with worms, we don't want to get into that right now. We're going to just stay with what the worm made.
Absolutely.
All right, so then bring me forward. So what's the plan going forward?
So it's working on both allergic and autoimmune diseases. And so we're really starting first with allergic disease. And where we are right now is we're going into clinical trials this year with our first molecule. And usually you have to start with healthy volunteers to get a baseline. So that's what we're doing this year, is we're understanding safety, tolerability. Also, there's this term called pharmacokinetics, which is when you give it orally, how long is it in your body and where does it go? What does it turn into? If it turns into this metabolite X, show us that it's being cleared out of the system. Typical FDA guidelines, FDA really cares about safety. So it's really understanding what is happening once we return this molecule back into mankind, which is actually pretty exciting if you think about it, because we used to have it.
Right.
We took it out.
Right.
And by the way, that's when allergic and autoimmune diseases suddenly surged, right? Is that a coincidence? So now we're returning for the first time a worm-derived molecule that I believe is intended to be part of the human immune system back into mankind for the first time this year.
We're missing a little section, which is you took this stuff that you isolated from the worm, you made it yourself, same exact stuff, and then you must have taken it up and down the road to see what it's doing to the immune system in cell culture or in the lab.
Up and down the road, yeah, I like that. Yeah, absolutely.
What did you find? Like, we found that it did this, it pushed things towards Th2, which is sort of dialing down the immune system. It did give us some of the guts of what it's doing for the immune system from what you
No pun intended. Yeah, so allergic and autoimmune diseases, sometimes people look at them and think about them as a seesaw, right? Like Th1 diseases happen to be, T cells, H helper, type 1, type 2. And Th1 cells, sometimes if you get more of a response of that, you can be more prone to getting autoimmune disease. And if sometimes you have a lot of Th2 cells, you can be prone to getting allergic disease. So originally we thought, maybe our molecule's doing, blocking a Th1 or blocking a Th2, but how is it working on both, right? Because the seesaw, how is it working? It's actually working rebalancing, sort of the triangle piece, if you will. So that's how it's able to help them both. And why that's so interesting to me is because in drug development, sort of what right now, one of the norms of drug development is just take something in the immune system and take it down, right? The steroids will just take something and take it all down. A monoclonal biologic, you've heard of them, the drugs that end with ab, like dupilumab, they find one thing and they'll take it down. And philosophically, I've always thought there's got to be a better way. Maybe we can work with the immune system instead of against it. Steroids are so harsh because it's taking everything down. And then these biologics are taking one thing down, but I don't know, it's not like the immune system is a set of dials, right? And nature always finds a way. You can take one thing down, your disease might find another way. So I think it's really compelling that the worms have evolved a technology where they're actually improving these rebalancing cells.
At the risk of getting into med speak, can you just go a little deeper about what you're finding when you study cells, when you study the immune system in the lab? Because you obviously haven't done it in people yet. You're about to do phase 1. Like, give an example of, like, we found that it does this to that.
Yeah, well, it's a block. In the setting of Th1 disease, we see that it's able to block those sorts of players.
Where and like how are you studying that?
So in an IBD mouse model, we could study an inflammatory bowel disease. It's like Crohn's disease and ulcerative colitis. You go into the crypts of the mice, you see all these neutrophils. Which is a type of granulocyte, means they're carrying chemicals and blowing them up. They're the ones that I told you that's like the police force where they come and they're like adding more holes to the wall, right? So the neutrophils. In allergic disease, those granules are called eosinophils. And so what we see in the setting of each model, that it can also reduce neutrophils in this setting and reduce eosinophils in this setting. And so we're really interested in exploring both, but what is this saying? Who is it? Leonardo da Vinci? Or how did he do everything? One at a time, right? So we're starting with an allergic disease, and then we're going to expand into multiple diseases after that.
That was great. So now you come back to I keep coming back to the philosophy, and we're so anthropomorphic, and where it's humans that are so important, and it's us. We got to get rid of the worms. We're going to get rid of the bacteria. We change our microbiome. We have fewer and fewer species. We know that there's consequences of that in the microbiome, different talk. But think about what she's saying here, which is that if in evolution, this complicated interacting immune system with lots of different players evolved to include something that the roundworm was secreting, and then we killed the roundworms because they're icky, and by the way, there are diseases. Hookworm can cause anemia and all sorts of other problems. So you understand why people did that. And now some important component, the Star Trek, the dilithium crystals. Something that's
you're
really
a Trekkie. I was going to go Star Wars.
Really important, and you suddenly take it out, and no more warp speed. So I find this so fascinating because it's a matter of looking at things. You turn a screw. My father-in-law used to love this expression. He would say, turn a screw 1/4 of an inch in your head on whatever the subject is, right? And I think about, I'm a gastroenterologist, H. pylori, Helicobacter pylori. We know that it increases the risk of gastric cancer, so you test positive for H. pylori, you kill it. Well, now we're finding out that it may increase the risk of Barrett's esophagus and esophageal cancer. By the way, the bacteria screw up the microbiome and cause all sorts of secondary problems. So it's not always intuitively obvious exactly what's going on, what you're doing with your, if it's living and in around our body and it's not us, let's get rid of it. Maybe not so much.
Absolutely. I mean, it's so funny that you use Star Trek as an analogy, because sometimes I use Star Wars as an analogy.
Okay.
Like the worms they're little Jedis. They have these little Jedi cloaking devices where it's like, don't come find me and get rid of me. There's nothing to see here. So really what we want to do is just use their tools. How can we borrow their tools and use the tools without the worms for our own purposes to benefit human health?
Let the pheromones be with you.
That's going to be the next T-shirt at your next merchandising table near you.
So we have 5 minutes until we go to T&A, but I'm wondering, I want to give you a chance to sum up, because I know there's going to be 1,000 questions here. At the end of the movie, still thinking about who's playing you.
Wait, Aquafina eating Reese's Pieces.
I like that. I'm all there. Give sort of a closing little spiel that sort of says, and so therefore, what I hope you took from today is this, and then let's open it up to Q&A.
Yeah, well, there's nothing better than may the pheromones be with you, but I think what I would love for you to take away with exactly what you said, it's hard for our intuitions sometimes to fathom. And if that's one message you could take away with you in general, even outside of today's session, I really urge you to take that with you. I mean, there's just so many things that, like, for example, Abraham Lincoln seems to me like a mythical figure. Things sometimes that we cannot see with our own eyes, like things before World War I, where footage, I just remember tanks in World War I and that's it. Things that we cannot see sometimes don't feel real to us, and I just want to simply urge you to reconsider that. Because back in the day, or before we were, in the civilization that we have now, hundreds of years ago, where people had worms in their guts, it was a normal part of their intestinal system and a part of their, if you will, their immune system, they didn't have as many of these diseases. Now we see people with Crohn's disease all the time, and autoimmune allergic diseases all the time. Everyone's complaining about eczema. You're like, oh, yeah, me too. Did you try this or did you try that? But there was a time where we didn't have that, believe it or not. So how can we return to that time? And how can we really value that? How can we value things that may not seem intuitive to you? And so I would just ask you to consider that, and having that lens when you view science and progress and possible innovation.
Great. So I've been pushing you a little bit more about specifics here. The journalist in me understands you have a company that is going to be getting funding. There are 10 people now, you told me, right? That you're going to be getting this round, that round, the other round. So we want to know, this is really exciting stuff. We're gonna see what happens going forward, and so we really want you to keep us, going, keep me in touch.
Yeah, we'll follow along.
What actually happens. But with that, we have 17 minutes and 20, 19, 18. Let's open up to Q&A. Yes, sir. We have a mic, or can you talk loudly?
I can talk loudly.
Oh,
yeah, that works.
So every night my children sleep with our dog, who is a descendant of a gray wolf, and along with my dog's microbiome, I guess. Why not go for the whole worm and have a domesticated worm in the future that could provide that whole, all those Reese's Pieces that worms produce?
You want to have a giant worm plushie that your kids sleep with at night?
Or a domesticated worm.
You mean Inu?
Inu, yeah.
He's saying, why go with the stuff the worm makes? Why not go with the worm? Because who knows? Maybe it's higher in fiber.
You could. So there's I know a guy in Mexico that's doing that, if you want to go there. I think that what I really, and you know what? I think that's a perfectly reasonable route to go. Okay, so if I were someone with serious allergic and autoimmune disease, and I know people with these, because people find me and they talk to me about this, they go find the worms, and may the worms be with them, right? I think what I'm really interested in is, what can I do in this world? What can I do with my background, with my medical degree and my scientific training? What I can do is I can really characterize something that they're secreting really well, and bring that, which I believe once we characterize something and the FDA is also giving its blessing that it's safe and that this can be distributed, it's orally available, people can have pills, I believe that's going to make the hugest impact, because it's something that then everyone will understand and that they can take and we can track and have data in clinical trials. So that's the reason, that's what I'm doing in my time in this world.
But keep an open mind, right? 200 years from now, they may be saying, can you believe they were only giving the molecules and they didn't realize that you give the whole worm? I mean, really, keep an open mind, which would be one of the themes here today. Again, I'm a gastroenterologist. We do fecal transplants for people who have things like C. difficile, colitis, right? And that's life-saving. Now it's a poop pill. It's a little bit less icky, but it's life-saving. If you had said 20 years ago, here's what we're going to be doing, people would have said, are you out of your mind? So it really is a good question. Yes, sir. And then we'll go over there and then over there. I knew there'd be a lot of questions.
Thank you. I'm a physician. My specialty was thyroid disease, and I basically made my living treating people with autoimmune diseases. And we know that many of these diseases are mediated by specific antibodies. Sometimes they're stimulating in the case of Graves' disease, sometimes they're inhibiting or blocking. But we also know that many families have these antibodies of different types that don't do anything. Are you suggesting that these small molecules may actually remove the antibodies, inhibit the antibodies, block their effectiveness? How is this all put together?
So I think that there are some worm molecules out there that people have characterized that work directly on, antibodies. But I think that our technology, at least our first program, is working on some of the regulatory cells that can then influence the B cells and the plasma cells that are then creating those antibodies. So where in that chain do you think the problem is originally? Is it in the genetic level? Is it in the regulatory level? Like if in those families that you're talking about, where does the disease, where is it starting, right? Is it in the gene of that B cell? Or is it somewhere post-transcript, like after the DNA code has been written to an RNA, to a protein? So I think it really depends on where that problem is. I mean, I don't know specifically which one you're talking about.
And one of the challenges, of course, is she's finding there's an effect, and we're trying to figure out the mechanism, or you're trying to figure out the mechanism in an extraordinarily complex system. So I think that'll be something that, how much other research is going on right now on this, aside from your company?
All over the world. Yeah, we're working with Rick Maisels, is considered one of the best in this field. And I mean, he was kind of like my rock star in grad school, right? Like that was the worm biologist, Rick Maisels.
Where is he?
In Glasgow. Uh-huh. He's working with us now, and we're actually having work being done in his lab as we speak. And so, we have a lot of really great minds that have come together to try to not just understand our first program, but also follow-on programs.
You doing anything in Africa?
Not yet, but I would love to.
You didn't finish my question. Are you anticipating removing these antibodies? Because the antibodies, in my mind at least, are what's causing the damage to the body.
I don't know that it's taking the antibodies and taking them down, because again, the technology that I'm describing is not taking something and taking it down. It's actually improving sort of like the police force, I don't want to use the word police force, but sort of like the regulating cells of the body that can maybe tell those cells, maybe don't make so much of those.
Or so maybe upstream.
Yeah, something upstream of that.
Of the antibody production. There was a woman, yes.
Thank you so much. I realize that this is in the very early stages, but realistically, how far away, how many years away do you think we are from seeing something that would be even a human clinical trial, much less something that would be on shelves or in pharmacies to be used as a product?
Yeah, well, our first human clinical trial is this year, and that's with healthy volunteers. And then, a year or two after that would be our phase 2 trial with patients, and then after that is the phase 3. So then you can see a couple years after that is FDA approval.
So all told, you're imagining?
It's like 5-6 years, we hope.
Have you come up with a name yet?
I'm open to names, so please send us any suggestions for names.
Okay. Yes.
I find it interesting you both are on stage because you work at NYU Langone, right? And you guys are Center of Excellence for Autoimmune Diseases, and you're trying to eradicate it. So,
You trying to
like,
put us out of business?
Yeah.
Well, you always have the empathy center.
No, but I'm just thinking, like, to the clinical trial and how fast can you go? I mean, as this is a monumental issue in America, and there's a lot of folks that come to places like NYU, I mean, how would you partner to try to speed up aspects of this so that those people can have relief?
Yeah, well, we can certainly work with clinicians there that can be part of the trials and help us to recruit patients. I mean, we really rely on clinicians and their connections to patients, not just because they are the ones that are helping to recruit them and overseeing the trial and making sure that everyone's okay, but they're also most familiar with the endpoint, meaning what is it that you want to know about our patient, how often they're, sneezing or how often you see a rash or whatnot. And on top of that, they trust clinicians as we expect them to. They're the gatekeepers that we as people trust. We go to them to ask them for their opinion. So I think we just want to work with them in understanding our story and our message and conveying that to people so that then people have an option to enroll or not.
And I was a computer nerd in another life, and I think about this the way you're describing it. You're not just saying, I mean, it's a big hypothesis, and we'll see what happens with it, right? But she's really talking again the trunk of the tree of changing the very interaction of and the way the immune system works at some level that's not yet understood. And it would be sort of like, going into a computer program and changing a line of code. In this case, it would be putting in this substance, and then you have to recompile, and you see, does it blow up? What happens? So there's a lot of road ahead in terms of figuring out as this works. So if it can make anything that's pharmacologically active and can help, it is pharmacologically active and can hurt, which is why you have this whole phase 1, phase 2, phase 3. So, but it's going to be fascinating to see.
No worms have blown up during this experiment.
Were any worms hurt in the making of this made-for-TV
hurt
movie?
in the making of this movie.
Okay, Peter Lee.
Incredibly interesting. And so I was just trying to imagine how you envision this drug. Let's say in 5 or 6 years we have this. It provides some sort of prophylactic benefit. And so 2 questions. Do you imagine that this is something you have to take every day, every week, every month, every year? And secondly, how do you measure the benefit? How do you, as a patient, as someone taking this, do you treat it like a multivitamin? What's the vision here?
Yeah, I think like I said, we want to reproduce as closely as possible to what the worm is doing. So I do envision it being something that you take every day. It depends on every profile. So if we're giving a molecule that leaves the system after 2 days, we would give it every 2 days, right? I think in our case, we would want to give it probably every day. And I don't know that I would think about it as a multivitamin. Yeah, I mean, we want to regulate it. Is the thing. Otherwise, I would have put it on the shelves of GNC already, right? But we just really want to carefully regulate and understand who it's going to and how they're using it. What was the other part of your question?
The question is, for something that has this type of prophylactic
measure
or preventative benefit, how do you know?
With every disease, we care about the endpoints that the clinicians, this is in other words the reason why we want to work with clinicians. They know the endpoints that are most closely linked to disease. And whether that's blood markers, but it's also patient symptoms, because sometimes you measure biomarkers from a person and you're like, oh, this is what you should look like. And they're like, that's not how I feel at all, though. Like, okay, you're saying all of my neutrophils or eosinophils are gone, but I still feel like crap and I'm still like sneezing or I still have indigestion. So we always care about the patient symptoms, and there's usually a patient scoring system, a questionnaire. So that's always a part of it.
But to be clear, it's not to give to everybody in the world. It's people with certain conditions, right? Initially, that's what you're thinking, not as something that we just take to avoid autoimmune problems in the future. And there's the half.
I don't know, maybe there will be.
I don't want to change your business plan. But anyway, there's the half-life of a substance, and then there's the biologic half-life. So you give something, it's gone through your system in 6 hours, but the effect's lasting. Do you have a sense of that with this compound or not yet?
Yeah, we do. Well, in our animal models, but now we need to see how that plays out in people.
So what's the half-life in an animal model?
So the half-life, well, depends on which molecule, but these molecules have a half-life of about a day to 2 days, and the effects can be as long-lasting. Well, it depends on what the effect is. It can last weeks, it can last even a month.
All right.
So I think that we would have to track. The best way to do it is you have your placebo group, or you then track them and you take them off of it. You don't, tell them and you measure and you ask. So, this is why I really care about using science to track all of those things. What happens when you come off of it? How fast will it return? Will you stay the same, or will you get even worse when you come off of it, right? It's like Ozempic, which, by the way, also came from nature, Gila monster secretion. When people stop taking that, then the effect comes back. It's not like you're reprogrammed after taking it once.
So if you can't get Ozempic because it's really in scarce supply, Gila we get. Where do you? Any other questions? Yes, sir.
Anupam Agarwal, I am a cardiologist. So very fascinating talk. I heard for the first time that worm can do that. What I was thinking when you gave the example of asthma. So asthma or diabetes, we have simplified that there is one etiology. For example, diabetes, we have divided into type 1 and type 2, and experts say there are multiple, etiologies. Similarly, in asthma, I believe we should study those patients who improved by treating with worms. Maybe they have a very specific kind of asthma. So it will not be applicable for all asthma patients. As you know, you may have treated many patients who had asthma as a child and that it goes away.
Sure.
There are so many variations in.
So you don't think this is going to treat every autoimmune issue?
You know what? It may, but I really want to just convey something really important that you were talking about, which is touching upon personalized medicine. So even with Crohn's, I said, IBD, you think about Crohn's and ulcerative colitis. I really do not believe that there are only 2 genres of that. And I think that with the advent of AI and a lot of the personalized medicine health apps, that we're going to learn all of that. So I'm really excited by this sort of a question, because I think that we really do need to understand these subtypes and help serve them. I think the challenges are always, you got to name your population. When you go into an FDA trial, it's almost like you've got to name like the 3-point shot you're going to make and like what statistic you're going to hit. And if you don't, then like it's over, right? So I think that, yes, we want to try to understand as best as possible what subtype. And one way that we're doing that is we're taking human PBMCs, like when a lot of...
PBMC meaning?
Peripheral blood mononuclear cells. So when you give blood, like patients that can go to the hospital, they can say, I've been diagnosed with this disease. You can give your blood and it's stored in a freezer. And then we can buy the blood from different subtypes and see how our molecule works on that, in that setting. Paired with a technology, for example, single-cell RNA-seq, which I won't go into great detail right now, but it allows you to see on multiple levels what is happening and truly understanding within your blood which of your blood cells are changing. So I think that this is a really important question, because as much as we're talking about how worms can, help the world, understanding at that level of integrity in science is really critical.
So we have 2 minutes left, and I'm gonna finish it up with a question, which is, at the end of clinical papers, there's a little thing that says limitations. Here was the limitations of our study. So what keeps you up at night? You're clearly excited about this. You're thinking maybe maybe it could treat a whole universe of autoimmune disease. But what are the limitations that maybe keep you up at night and think, I'm worried about this?
Limitations of the worm molecule, or things that could keep me up at night?
Yeah,
Andrea?
no, I don't
It's
mean
not
about
the worm
the
molecules.
whole chocolate peanut butter thing, but the conundrum. But you're a researcher, MD, PhD. You're in a serious lab with serious people. And you're painting a story which is really exciting, and of course my job is to sort of say this is exciting, but we have to think about the steps that. So let's trust and verify, and I'm excited about it too. But what's the buts for you? What do you think? Like, are there things that you're thinking, well, I hope, but it may turn out that's going to happen, or I'm worried about this, or is it just full speed ahead and
Oh, it's
damn
never
the
just false.
torpedoes?
We never damn the torpedoes. I think that look, this gentleman kind of nailed it. Subtypes, who's going to benefit from this? My nightmare is we go in, we treat a population, it helps some of them, but we didn't name exactly that population or that statistic, and then we don't get the chance and the opportunity to find where it can help, and also the vast potential of other molecules and other diseases we can treat. I think the potential is so great here, but it's all about the challenge of knowing what population you're going to go into, what statistic you're going to see, what endpoint. So it's about picking the right first fruit, right? You ever read The Bell Jar, Sylvia Plath? She talks about how she's sitting under this fig tree and she's like, oh my gosh, I don't know which one to choose. And then they just drop in front of her. And she's like, now it's too late, I can't eat any of them. But I guess that would be my Sylvia Plath nightmare.
And I think this is the first talk here ever to end with the 3 words, Sylvia Plath nightmare. So with that, thank you very much. This is fantastic work.
Thank you. Thank you.
Well
Thank you
done.
so much.
Yeah, well done. Great.
Sure.
Sorry.
I'm so sorry. Thank you. Let me get off of this pedestal. Let me get my mic off and then I'll come.